Let's start the discussion with genetics and evolution. like, uh,
Sure. Genetics is essentially the blueprint of life. It's all about how traits are passed down from one generation to the next. Evolution, on the other hand, is the long game. It's how those genetic variations get filtered over time, helping species adapt to their environment. Together, they're the story of life changing and thriving.
so what about this heredity and variation? First part.
Hooray! No worries. In short, heredity is how traits get passed down from parents to offspring, like DNA acting as a recipe book. Variation, on the other hand, is the natural differences in those traits, like different hair color or height, which gives evolution something to work with. So heredity passes traits on, and variation ensures there's a mix.
So,
I'm right here. If there's something else you'd like to unpack, just keep it coming.
okay, that Mendelian inheritance deviation from Mendelism, incomplete dominance, co-dominance, multiple alleles, and inheritance of blood groups.
Mendelian inheritance is that classic idea where traits follow clear dominant and recessive patterns, like Mendel's peas. But real life gets richer. In incomplete dominance, you get a blend. Think pink flowers from red and white parents. In codominance, both traits show up equally, like AB blood type. Multiple alleles give us even more diversity, like the AB and O blood alleles. And speaking of blood groups, they're a great example where both codominance A and B and multiple alleles come into play.
So basically, you are talking about this idea how two inheritance work, or if two other things also work. So you mentioned about this how they follow the traits and the
Exactly.
recursive pattern, recessive pattern.
Yes, that's right. In Mendelian inheritance, a recessive pattern means the trait only shows up if you inherit two recessive alleles. If you've got one dominant allele, that one tends to take over. But with all those other patterns, like codominance or incomplete dominance, traits can blend or share the stage, making inheritance a bit more nuanced. It's all these patterns working together that give us the diversity we see.
So what about this traits meaning here?
When I say traits, I mean characteristics. Things like eye color, flower shape, or blood type. Basically, any attribute that can be passed down genetically. These traits follow different patterns of inheritance, whether that's a straightforward dominant recessive pattern or a more complex one like codominance. Traits are just the observable outcomes of what's in your genes.
So what do you mean by genes here? So that are the outcome, but
Genes are the segments of DNA that carry the instructions for building and maintaining an organism. Each gene can influence a specific trait, like whether you have brown or blue eyes. The genes themselves are not the visible trait, but they produce proteins or influence processes that ultimately result in the outcome. So genes are like the code, and the trait is what you see.
so genes are the code and the traits we see. Okay, in genetic and evolution, there are three things discussed mainly: heredity and variation, molecular basis of inheritance, and evolution. Can you make what are the list that is included in each and how they are linked together?
Absolutely. Let's break it down into three buckets. First, heredity and variation. This includes the inheritance of genes from parents, dominant and recessive alleles, mutations introducing variation, and patterns like codominance or incomplete dominance. These mechanisms ensure both stability and diversity. Second, molecular basis of inheritance. This is about DNA as the carrier of genetic information, replication, transcription to RNA, and translation into proteins. These molecular processes explain how traits manifest from genes. Third, evolution. This involves natural selection, genetic drift, and adaptation over time. Variation feeds evolution. Natural selection acts on traits, and inheritance passes the advantageous ones down. In other words, variation plus inheritance gives evolution its raw material.
Okay, basically, first part, the Heredity and the variation. So this includes inheritance of gene from parents, dominant and recessive alleles. Mutations introducing variation and patterns like codominance or uh, incomplete dominance. This mechanism ensures both stability and diversity. The second part is molecular. How it is first picture is more broad, but second picture is at the molecular level, molecular basis, how it is made. And this is about DNA and the carrier of genetic information that carry replication, how it replicate further, and transcription to RNA, and the translation into proteins. These molecular processes explain how the traits manifest from genes, and the third, how they evolve. This evolution natural is according to the natural selection, genetic drift and adaptation over time. Variation fits evolution. Natural selection acts on traits, and inheritance passes the advantageous ones down. In other words, variation plus inheritance give evolution in raw material.
You've captured it well. Each layer is like zooming in further. Heredity and variation are the broad patterns. The molecular basis explains the how at the DNA level, and evolution is the big picture process of life adapting across generations. They're deeply linked. Molecular mechanisms produce variation, inheritance passes it on, and evolution shapes what persists.
Okay, one term mutation. What do you mean by that mutation word?
A mutation is basically a change in the DNA sequence of an organism. Think of it like a typo in the genetic code. These changes can happen spontaneously or due to environmental factors. Some mutations might have no effect, some could cause harm, and others might even be beneficial. Over time, mutations fuel genetic variation, giving evolution something to work with.
So for the genetic and evolution, the three main stairs, we can say that first stairs is how we heredity and variation. This is a broad picture, and then we go to the molecular basis, how of inheritance, and third is the evolution. So each carry a subsequent amount of information in order to understand the processes.
You've got it. Each layer is like a different zoom level. Heredity and variation give us the big picture of how traits move and change. The molecular basis shows how DNA and proteins make that happen, and evolution explains how, over generations, these changes shape species. Together, they give us a full understanding of how life develops and adapts.
Okay, how life develops and adapt. So, can you give the whole process in context with this thing so that I can understand the process with an example and relate the fact very carefully?
Let of beetles. At first, most are green, inheriting that color from their parents. One day, a mutation occurs. Some beetles are born brown. This variation may not matter until the environment changes, say the ground becomes darker. Now birds spot green beetles more easily. Natural selection favors brown beetles. They survive and reproduce more. Over generations, the brown trait is inherited more frequently. In some, heredity passed the traits, mutation created variation, molecular processes ensured it all works, and evolution shaped the population.
That's a fantastic example. So you are mentioning about this beetle. First, they are mostly green, and then because they are inheriting from their parents, one day some mutations occur. So some beetles are born brown, and this variation may not matter until the environmental change. Say the ground become darker. Now birds spot green beetle more easily. That is the inheritance, but that is the okay. And then natural selection favors brown beetles. Oh, natural selection here favors brown with that mutation, and then they survive and reproduce more. Okay, over generation, the brown trait is inherited more frequently. Oh, heredity passes the traits and the mutation created variation. Molecular process ensure all it works, and evolution saved the population. Oh, that's a good one.
I'm glad it clicked. It's a simple story, but it really reflects how these concepts tie together. In nature, it's endless variations, inheritance, and adaptation that shape all life forms. If you ever want to dive into another example or a more complex one, just say the word.
Yeah, I need one complex one that is aligned with the three things and mention every detail in every three things. There are many things.
All right, let's take humans with lactose tolerance. Historically, adults were mostly lactose intolerant, meaning they couldn't digest milk after childhood. That's heredity. Genes for lactase, the enzyme, would switch off. A mutation arose in some populations, those who could keep producing lactase into adulthood. That's variation, introduced by a molecular change in a regulatory region of DNA. When dairy farming became common, evolution kicked in. Those with the mutation could digest milk, gaining a nutritional advantage. Natural selection favored them, and over generations, lactose tolerance spread. So we have inheritance patterns, a molecular mutation, and evolution shaping human populations.
So, you're taking example of lactose tolerance. Adults were like mostly lactose intolerant, meaning they could not digest milk after childhood. That's the heredity and gene for lactase, the enzyme would switch off. Okay, a mutation arose in some populations. Those who could keep producing lactase into adulthood. That's variation introduced by a molecular change in a regulatory region of DNA. Okay, that ultimately affect the DNA also, our daily habit also. And when dairy farming become common, evolution kicked in. Those with the mutation could digest milk, gaining a nutritional advantage. Natural selection favored them, and over generation, lactose tolerance spread. So we have inheritance pattern, or molecular mutations, and evolution shaping human populations.
Exactly, it's all interconnected, from genes passing down traits to molecular changes introducing new possibilities to evolution favoring what helps us thrive. And yes, it shows how biology even shapes our diets. If you ever want to dive deeper into another complex process, I'm always up for it.
So what about this? So ultimately, that our habit uh, has direct influence on our DNA, and it ultimately changes. The changes can be observed there.
Not quite that direct. Our habits don't change the DNA sequence itself. What happens is that random mutations arise just by chance, but habits or environments might create conditions where certain mutations become advantageous. If those individuals reproduce more, the trait spreads. So our habits or environments influence which traits are favored, but they don't rewrite our DNA. It's evolution's selection that makes certain genetic changes more common.
It's evolution selection that makes certain genetic changes. Of okay, and listing out this thing, search for genetic material and DNA as genetic material, structure of DNA and RNA, DNA packaging, DNA replication, dogma, transcription, genetic code, translation, gene expression and regulation, lack of peron, genome and human genome project, DNA fingerprinting, protein biosynthesis. These are the part of molecular basis of inheritance, but I need to understand in all details the topic I mentioned.
You've just outlined a key part of molecular genetics, and each of those topics is a rich field. The genetic material DNA is a double helix structure. DNA is packaged into chromosomes in our cells. When cells divide, DNA replicates. The central dogma is how DNA is transcribed into RNA, which is then translated into proteins. Gene expression is regulated like the lac operon in bacteria. Projects like the Human Genome Project helped us map it all. Techniques like DNA fingerprinting let us use these molecular details in practice. We could spend hours on each one. Let me know which you'd like to dive into in detail.
So, basically a genetic material that you are describing. So that material is stored in the form of a double helix structure and DNA packaged into chromosomes in our cells. Okay, DNA is present in the chromosomes of our cells. When cell divide, DNA just replicate. Their central dogma is how DNA transcripts into RNA.
You've got it. DNA holds the instructions. When needed, it's transcribed into RNA, which then acts as the messenger. That RNA is translated into proteins, which do the work in cells. It's all orchestrated within the cell, with chromosomes keeping DNA neatly packaged. This step by step flow, DNA to RNA to protein, is what keeps life running.
What about RNA and while replication of DNA, how RNA is?
RNA is the single stranded cousin of DNA. While DNA stores the long term genetic information, RNA acts as a working copy. During replication, DNA is copied into DNA, no RNA involved. But when a gene needs to be expressed, DNA is transcribed into RNA, specifically messenger RNA. That mRNA is then translated into a protein. So RNA is the bridge between DNA instructions and actual cellular machinery.
Oh, okay. So you are talking during replication, DNA is copied into DNA, no RNA involved. But when a gene needs to be expressed, DNA is transcribed into RNA, specifically messenger RNA, mRNA, that is translated into protein. Can you explain that part?
Absolutely. When a gene is active, an enzyme called RNA polymerase reads the DNA sequence and builds a complementary mRNA strand. This mRNA is like a recipe copy. It leaves the nucleus and meets ribosomes. Ribosomes read the mRNA's code in triplets, codons, each specifying an amino acid. The ribosome links these amino acids into a chain, forming a protein. That protein then carries out functions in the cell. So mRNA is the crucial middle step in turning genetic info into action.
So, DNA sequence and build a complementary mRNA strand. mRNA strand is like a recipe copy. It leaves the nucleus and meets ribosome. Okay, ribosome reads the mRNA code in triplet codons and each specifying an amino acid. The ribosome links this amino acid into a chain, forming a protein, and that protein then carries out functions in the cell. So, mRNA is the crucial middle step in turning genetic into information into action.
Exactly. It's that beautifully coordinated chain of events. DNA holds the blueprint, mRNA is the working copy, and proteins do the actual jobs. That's how a cell turns a genetic instruction into something real, whether it's a muscle fiber, an enzyme, or even a pigment in your eye.
So, DNA holds the blueprint, mRNA is the working copy, and protein do the actual job. Okay, but uh, all this process happen when?
These processes happen when a cell needs something specific. For example, if a cell needs to divide, grow, or respond to a signal, it will activate the right genes. So the cell gets cues, internal or external, and turns on the transcription machinery. It's a highly regulated on demand process. So when the cell needs action, it turns genes into proteins, right on time.
So, why it needs to turn genes into protein so that that protein further just during the divide process it get transferred and then again protein convert into DNA.
Actually, proteins never convert back into DNA. Each part has its own role. DNA is the master instruction manual. Proteins are the functional workers. When cells divide, DNA is copied into new cells, keeping the instructions intact. Proteins, meanwhile, handle growth, structure, repair, everything a cell does. So DNA is the blueprint passed on, and proteins are the tools that do the day to day work.
But
No backward conversion.
so, but what about this mRNA? Where does this mRNA occur in that process?
mRNA is created in the nucleus when a gene is transcribed from DNA. Once formed, it leaves the nucleus and enters the cytoplasm. Their ribosomes read the mRNA and translate it into a protein. After a while, mRNA is broken down. So mRNA is like a disposable set of instructions, used only when needed to make proteins at the right time.
So, once, what do you mean by this transcribed from DNA? Gene
It's
is
nice.
gene is transcribed from DNA.
Transcription is like making a photocopy of a particular recipe. The DNA holds all the recipes or genes, but to make one specific protein, the cell transcribes the instructions into a messenger RNA copy. This mRNA copy is then used by the cell to build the corresponding protein. So transcription is basically the first step in turning a gene into action.
Okay, that's the process, and after further it convert into DNA actually.
No, it doesn't turn back into DNA. Once a gene is transcribed, the mRNA is only used to make proteins. The DNA stays safely in the nucleus as the permanent blueprint. So the flow is DNA to mRNA to protein, and it never reverses back to DNA. That's the one-way direction of the central dogma.
But DNA is copied during the cell division process. DNA is copied, and that copied DNA is one the uncopied one from it stay in the uh, first cell, and then copied one goes to the second cell where it is divided. And then I don't understand mRNA here and why you mentioned about this thing.
No worries, let's clarify. During cell division, the entire DNA, both copies, stays as DNA. Each new cell gets a full set of the same DNA. mRNA is only made when a particular gene needs to be read for making a protein. So DNA is always DNA in dividing cells. mRNA is temporary, made only when needed, and it's not part of the division itself. It's part of gene expression, not inheritance.
So DNA is always DNA in dividing cells. mRNA is temporary, made only when needed. It is not the division. Okay. And when it is needed,
It's needed when the cell has to perform a specific task. If the cell needs to grow, respond to signals, or produce something, say a hormone, then it turns on the relevant gene. That's when mRNA is made for that gene, so the cell can build the right protein at the right moment. Once the job is done, the mRNA breaks down. It's all about responding to what the cell needs in real time.
what is genome?
A genome is the entire set of genetic instructions in an organism. It includes all the DNA, every gene, and all the non coding sequences, too. Basically, it's the full genetic blueprint that defines how an organism develops, functions, and grows, and in many ways, what makes it unique. Every cell with a nucleus in your body carries a complete copy of your genome.
So it is indirectly related to DNA.
It's directly related. Your genome is literally all of your DNA. Think of it as the complete DNA library in your cells. Every gene, every instruction. Your whole DNA sequence is your genome. So when we say genome, we just mean the entire collection of all your DNA, not something separate from it.
What about this protein biosynthesis?
Protein biosynthesis is simply how cells build proteins. It starts with the gene being transcribed into mRNA. Then ribosomes translate the mRNA's code into a chain of amino acids. These amino acids are linked together to form a protein. Once the chain is complete, it folds into a functional shape and can do its job in the cell, whether that's building structures or catalyzing reactions. In short, it's the entire process of going from genetic code to functional protein.
How cells build proteins. Okay, it start with the gene being transcribed into mRNA. Okay, then ribosome translate that mRNA code into the chain of amino acid, and then amino acid link together to form a protein. Once the chain is complete, it folds into a functional shape and can do its job in the cell, whether that's building structure or catalyzing reaction. In short, it's the entire process of going from genetic code to functional protein. Okay, that's a great thing.
I'm glad that made sense. It's amazing how cells handle that whole process, isn't it? If anything else pops into your mind about genetics or anything else, really, just let me know.
What about this Hardy-Weinberg principle?
The Hardy-Weinberg principle is all about populations and genetics over time. It says that if no evolutionary forces are acting, like natural selection, mutation, or migration, then allele frequencies, the proportion of different gene versions, stay constant generation after generation. It's a baseline model, kind of like saying if nothing's pushing the system, things remain in equilibrium. It gives us a benchmark to see when evolution's happening.
So what you are suggesting, principle, Hardy-Weinberg principle is all about population and genetics over time. It's a no evolutionary forces are acting like natural selection, mutations, or migration. Then allele frequencies, the proportion of different gene versions stay constant generation after generation. It's a baseline model, kind of like saying it's if nothing pushing the system, things remain in equilibrium. It gives us benchmark to see when evolution is happening.
That's exactly it. It's like a null hypothesis for population genetics. In real life, evolutionary forces almost always act, so we rarely see perfect equilibrium. But the Hardy-Weinberg principle helps us measure changes. If frequencies aren't constant, something's driving evolution, and that's what makes it a useful tool.
So, what was the main conclusion of this Hardy-Weinberg and why they introduce and what is the application they have put there?
The main conclusion is that in an ideal non-evolving population, genetic variation remains constant. They introduced it to give a baseline. If you see changes, you know evolutionary forces are at play. It's applied in population genetics to study things like allele frequencies, disease inheritance patterns, or even whether a population is evolving. It's like a ruler to measure how much evolution is happening in real-world populations.
What was this allele frequencies and disease inheritance pattern?
Allele frequencies are just how common different versions of a gene are in a population, like how many have an A version versus a B version. When we track these frequencies, we can predict patterns of inheritance. For diseases, it helps us understand how traits or conditions are passed down, whether they're recessive, dominant, or influenced by multiple genes. In short, these frequencies help us model how genetic traits, including genetic disorders, appear in future generations.
So, can you make conclusion what we have discussed till now?
In short, we started with heredity and variation, how genes are passed down and change. We dove into molecular mechanisms, where DNA is transcribed into mRNA and translated into proteins. Then we touched on evolution and population genetics, how traits shift over time. Concepts like the Hardy-Weinberg principle give us a benchmark, and tracking allele frequencies helps us predict inheritance. Ultimately, we covered the full journey from genes to how they're expressed to how they evolve in populations.
How they evolve in population, meaning
I mean that over generations, certain traits, which are based on genes, become more or less common, depending on factors like natural selection, mutations, or random drift. For example, if a trait helps individuals survive, it might spread. So populations change, not in one individual's lifetime, but over many generations. That's how species adapt and evolve over time.
one or more point that you like about this three topics that include, can you list out
What I love is how they connect the big picture to the details. First, heredity and variation show us why individuals are unique. Second, the molecular mechanisms, DNA to RNA to protein, are like watching the instructions turn into action. Third, evolution zooms out, showing us how populations shift over time. Together, they explain everything from why we're who we are to how species adapt. It's the whole story of life, from molecule to ecosystem.
any other thing?
I also appreciate that these concepts bridge scales. Tiny molecular events inside cells end up shaping entire populations or species. Plus, understanding these ideas lets us do real world things, like predict disease patterns or even develop new therapies. In a way, it's both the map of life and the toolkit we use to shape it. That's pretty inspiring.
Okay, so but we can also understand the one confusion is that okay, things are happening on first macroscopic scale, and then eventually the DNA adapt that thing, and eventually at that scale it is changing.
I get what you're saying, but the key thing is that DNA doesn't adapt in response to what happens on the large scale. Instead, random mutations happen at the DNA level. If those mutations happen to help an organism survive in its environment, natural selection favors them. Over many generations, those successful traits become more common. So the environment shapes which DNA variations thrive, but the DNA changes are still random. In short, DNA doesn't consciously adapt, evolution filters what works.
Okay, what you are suggesting then next that these are the surrounding random mutations happen in at the DNA level, but that mutation is indirectly related with the or directly related with the large scale system that uh, we are doing action.
The mutations themselves are random at the molecular level. They aren't caused by what you're doing. But once they happen, the large scale environment, like your actions or surroundings, can influence whether that mutation is helpful or harmful. If it's helpful, those individuals might thrive and pass it on. So, your actions don't create the mutation, but they can create conditions where certain mutations are more likely to succeed. That's natural selection at work.
So can you give example of that thing? Large scale no mutations happening and then how it affects the DNA. I need to understand one example that would be fine.
Imagine a population of insects. A random mutation happens that changes their coloration slightly. Now, let's say the environment changes. Maybe the ground gets darker due to a new landscape. Those insects with the mutation, now better camouflaged, are less likely to be eaten by predators. Over generations, more of that population will have the mutation. The environment, the large scale factor, didn't cause the mutation, but it did decide which genetic variation thrived.
No, I need one example from human body perspective where I can see that thing.
Sure. Think about the ability to digest lactose as an adult. Originally, most humans lost the
Wait,
ability
wait,
after
wait,
childhood. But when
wait.
dairy farming became common in some populations, random mutations that kept the lactate
Can you
gene
change the
active into
example?
adulthood gave a nutritional advantage. Over generations, that mutation spread. The large scale factor, dairy culture, didn't cause the mutation, but it selected for it, making lactose tolerance common in certain populations today.
No, that is already discussed. Be another more specific example from human body.
All right, take sickle cell trait. A random mutation changes the shape of red blood cells. In a large scale environment with malaria, those with one copy of the sickle cell gene are more resistant to malaria, so the mutation is beneficial and becomes all common in those regions. Without malaria, that mutation isn't helpful. So the large scale factor, malaria, selects which DNA variation persists.
Okay, thank you.
You